Hello, everybody, and welcome to our webinar tonight presented by the Children's Tumor Foundation, The Path to Approved Treatments for NF2 schwannomatosis. My name is Kate. I work with CTF, and I'll just be our host this evening for what we're about to hear. I want to quickly mention, and I'm putting this in the chat as well, that there are closed captions available tonight. Just give me a moment. While I put that in here. Excellent. So you can turn on closed captions. They're available with a live captioner by clicking on the CC button at the bottom of your Zoom screen. You can adjust the size of the captions as well by selecting caption settings. So we welcome everyone to introduce themselves in the chat, where you're from and your NF connection. And please try to put your questions for our presenters tonight in the Q&A. This is a webinar and not a meeting, and so if we can keep questions in the Q&A, that will really help to keep us organized and make sure that we see the questions that are coming in. But if you want to introduce yourselves and say where you're calling in from and your connection to NF in the chat, you're more than welcome to do that. You can also use the upvote feature in the Q&A tonight to nominate a question that you would really love to be asked live. I'll be doing that myself at the end of all of our presentations this evening. So without further ado, I'm pleased to introduce our three speakers for this evening.
Kate,
yes.
There is a problem. I see multiple people saying that the chat is disabled.
Yes, I will fix that as soon as you start, Annette. I apologize about that, guys. It's just a setting that I need to change. So thank you. So let's get started. So tonight we have three people who are going to act as our guides along this journey to approved treatments. Dr. Annette Bacher is the president of CTF, and she'll be talking to us tonight about where this all started and how our earliest efforts have gotten us to where we are today. She's a global disruptor, has a PhD in biochemistry, and has leadership experience in the academic and pharma and biotech research enterprise in Italy, Belgium, France, and the USA. Following her, Dr. Scott Plotkin serves as the chair of the CTF Clinical Care Advisory Board, is lead investigator for the NF2 INTUITT trial, and a professor of neurology at Harvard Medical School and the director of cancer neurology at Massachusetts General Hospital. Dr. Plotkin's research focuses on developing clinical trials for patients with NF1 and schwannomatosis. He has served as principal investigator on single and multi-center clinical trials and is an active member of several national consortia. Dr. Plotkin will focus on where we are and what we know today. And lastly, Dr. Bridget Gerlich is CTF's new Chief Medical Officer. Dr. Gerlich is an accomplished pediatric oncologist, scientist, and senior-level pharma biotech executive with extensive experience in the healthcare landscape, spanning basic research, patient care, and global drug development. Her prior experience includes 10+ years as a physician scientist at a large academic medical center, followed by 15+ years in oncology clinical development in both global pharmaceutical corporations and biotech organizations. Dr. Gerlich joins us tonight to look to the future and see how the work that you're supporting through CTF will get us there. So I will make those corrections to the settings for the chat and the Q&A. And without further ado, I welcome Annette. Thank you.
Good evening, everybody. I'm the historian of the night. So I'm going to walk you through a couple of slides, like what have we been trying to do? And then I will hand it over to Dr. Plotkin to tell us more about that crazy, amazing platform trial. So with that, let me share my screen with you. And so, okay, so as Kate announced, we will be talking about innovative solutions to accelerate the path to approval. So the first two slides are on purpose a little bit on the dark side, because this is how the world looks outside CTF. You have an idea, you apply for funding, you wait, you do the experiments. The world waits for the publication to come out, then you test in cells, then you wait for the publication, and so on and so on and so on. And as you can see, this is a very long process. And what we have understood as CTF is that what can we do to shorten this and to make this a little more condensed and to get faster to drug approvals? To make it even darker, For rare disease, this is even worse, right? We have few patients, so you need multi-site clinical trials, which is that many sites have to agree to run the clinical site together. That leads sometimes to long recruitment times and long negotiations. And on top of that, rare diseases such as NF and schwannomatosis are very often, unfortunately, progressive and heterogeneous and also multi-organ. So you can imagine that you're looking at different manifestations, different ages. So even the protocols, the clinical protocols are complex. So that's the dark side. Let's now go to the light side. What have we done? What has your investment done to accelerate the process? Providing funding to increasing the review speed. How quickly can we get these reviews done? Less than a year. The NF data portal, instead of sharing only your data after the publication, which sometimes can take a year between the moment you discover and the rest of the world sees it, the community has agreed that it would be a good idea that CTF launches the NF data portal, which has now really become a standard, and a lot of other funders such as NTAP, CDMRP, the Gilbert Foundation, the NF Research Initiative, everybody is now agreeing and incentivizing their researchers to share their data on the data portal. One of the things that I will mainly talk about tonight is how can we improve and accelerate the preclinical processes, evaluate treatments in cell and in animal models faster, and bring multidisciplinary teams together. And then the last part, which is mainly going to be Dr. Plotkin's part of the show, is how can we accelerate clinical trials with these platform basket trials that Dr. Plotkin will talk about in a minute. So how did it all start? In 2008, the foundation started something that was called the NF Preclinical Consortium, which was a very interesting consortium or group of labs, multiple labs that came together that had animal models. As you know, in NF, we have different manifestations, and these animal models had different manifestations. And instead of testing a drug one manifestation at the time, with each time the time in between to get the information published, they decided and they agreed that it would be great to test drugs in multiple animal models at the same time and share the data. And that has been super valuable. They tested over 95 drugs over about 8 years, and they have collaborated. And you know what? That has led to the approval of Koselugo. So one of the labs, or the labs, had shown that MEK inhibition, which was not exactly with Koselugo, but that MEK inhibition actually shrunk plexiform neurofibromas significantly, and that had created the whole energy of activities that has led to the approval of Koselugo. What we do know, though, is that animal models are expensive and they're rather slow because, as you know, tumors in NF fortunately grow slow, but that also means that the time to run an animal model can sometimes lead up to a year. And you can only test very few treatments because it's low and it's expensive. The second thing is what we learned is that it would really be great if the clinicians and the clients who are the patients, that they would be in the room to make sure that we're working and that we're investing in the right treatments, and not that we wait until the last moment and that then the clinician or the patient says, this is a drug that I'm not interested in. And then, which is a third challenge that we were still remained with, is that sometimes negotiations between academia and pharma can be challenging. And I can talk about that much longer, but I'm not going to do that. So what we decided was we launched SYNODOS in 2014. Who are these people? Multidisciplinary, as you can see. You see here our favorite clinicians, Dr. Plotkin with a beard, Jayshree, the patients, Tracy Galloway, CTF was there, Salvo and myself were there, then a lot of researchers were there as well. And what is this? This is not a graph that I want you to completely understand, but I want you to embrace for a second the complexity of this study, where normally you would do cell screen, animal screen, and then you go into people. And what you can see here is that the yellow boxes are cell screens. The violet boxes are the animal models. Those are the in vivo screens. And the green is like an extra. Not only are we testing these drugs and these treatments in cell models and in animal models, which, as you can see, it's not sequential, it's at the same time, and things are combined and iterative. But we're also understanding, for some of you may have heard about kinome profiling, transcriptome, exome, proteome. So all these omes are also here. So not only are we screening drugs, we also understand why they work or not work. What has that given us? What has that learned us? We tested treatments in multiple animal models. That was kept from the NF preclinical consortium experience or NF therapeutic experience. The second was we want to increase the number of treatments that were evaluated. So what happened, what was the improvement that SYNODOS brought to the table is that the cell and animal models were included as part of that same big group consortium. And then clinicians and patients were together with the researchers. So we had that multidisciplinary aspect. What still was open was that the negotiations between academia and pharma are still sometimes challenging, and that will lead to the launch of the preclinical hub that Dr. Garlick will talk about later. Just a minute, and I don't want to steal the show here, but I think this is what Dr. Plotkin will teach you about a lot in the upcoming presentation. But this is this fantastic INTUITT trial, where you look at a platform, where you look at different drugs, and where you also look at the basket. And the basket is you're looking at multiple manifestations. The great thing here is that we have seven sites that are participating. There's 2 drugs that are already in trial, and we have more slots, and we know which drugs. The only thing that is holding us back for the moment is the cost. We have to count. We counted it when we measured it together with Dr. Blakely and Dr. Plotkin, and it's about $2 million per trial for 40 patients in 1 year. So basically in 1 year, pharmaceutical company will know whether a drug has hope or not, has a signal or not. So now let's go back to our dark slide and let's see if we can make this a little lighter. If you add the funding component, as I said in the beginning, these 2 go away or are significantly reduced in time. What did the data portal do? You stop waiting for publications to come out because researchers and clinicians are willing to share their data earlier. The NF preclinical consortium brought all these animal models under one roof. So instead of one at a time, now we have multiple animal models under the same roof. SYNODOS also included the cell testing, and the platform trial, as you will learn from Dr. Plotkin, will allow us to test multiple treatments under the same roof again. So needless to say that all the investments that you have made in the foundation over all those years have really been aimed at reducing the times and reducing the complexity of drug evaluation and bringing the right drugs to the clinic. So with that, I'm going to thank you. Help us shorten that path to approval by joining our movement. And with that, I'm going to hand it over to Dr. Plotkin.
Wonderful. Thank you, Dr. Bakker, for the great talk. Let me see. First, I have to share, right? Oops. So you have to disable my closed caption. Sorry about that. And I will restart it. I will look to see if I have the right. Let's see. Is that the correct? That sums up. Okay. Well, thank you for the opportunity to be here. I love scanning the chat and seeing a lot of my friends on there. And thank you for being here and for everyone who's making the time to be here today. I want to pick up this thread and give you a little update about where we are now in testing brigatinib for progressive tumors for persons with NF2-related schwannomatosis. And I want to emphasize here all the site PIs like me who have participated, and the many, many other people whose names will be at the end of the talk as well. This has really been a team effort. First, some disclosures, and this is so important for everyone to understand that I am showing you preliminary results. And like all clinical trials, we continue to do statistics. And so please look for updates. We should not anchor on the exact numbers I show you today, but they're going to be pretty close to the final numbers. Second of all, of course, to just acknowledge the support of Takeda Children's Tumor Foundation for this trial, pivotal in making it happen. And then finally, my own disclosures, which at Harvard we have to disclose during all of our talks so that people can evaluate those. I want to start off with an overview for everyone here about the drug development process. And this is a figure that is taken from a publication by the U.S. Food and Drug Administration. And what it shows is, if you think about drug development in terms of preclinical research in a laboratory, followed by clinical studies in humans, and then followed by a review at the Food and Drug Administration. This is an example of the US procedure for those who are not living in the United States. And then what I want to emphasize here is, after a period of preclinical research in which thousands or hundreds of thousands of molecules are examined, a small percentage of those get advanced in the clinical studies, and there's overlapping studies of safety, which we call phase 1 studies, efficacy or how well a drug works, phase 2 studies, as well as phase 3 studies, where we compare a new drug to an established treatment. And you'll see that these are overlapping, but leading ultimately to review of what we call an NDA, a new drug application to the FDA, and hopefully approval. And so this is both a longitudinal and an overlapping process. What is important to recognize, and we do here, is that developing new drugs is risky and expensive. So, for example, in this publication from Tufts, which is one of the premier economic evaluators of the drug development process, they looked at a series of new drugs between 1995 and 2000, 1,400 drugs, and were trying to estimate the cost of development. First, they looked at what was the chance that a drug would be advanced from the phase 1 to the phase 2 process. And that was about only 60% of drugs that started in phase 1 made it to phase 2. Only 35% of drugs that started in phase 2 made it to phase 3. 62% of drugs made it after phase 3 to a submission to the FDA. And of those, the chances are good, 90% of those actually got approved. But when you put all this together and say, what percentage of medicines that start in phase 1, which is only a tiny fraction of what's studied in the lab, what proportion of those actually make it? Only about 12% of medicines made it all the way to approval. So how much does it cost? I'm giving you this big funnel where you start broad and end up with a few drugs. If you look at the pre-human development costs and then the human development cost and a total, this is in millions of dollars in 2013. The total cost of developing a drug is estimated in 2013 to be $2.558 billion. An incredibly expensive process. And for that reason, this is, of course, very daunting. So we're trying to repurpose drugs, avoid this, and have high-risk shots on goal so that we can get drugs approved more quickly and for a lot less money. And it's quite a challenge. Here is where we've been. This is a list of trials that have been completed for NF2-related schwannomatosis. On the left are the actual drug names. These are the phase of the studies. These are the molecular targets. These are the number of patients. You can see anywhere usually from about 5 to 25 patients. These are the age groups, predominantly for adults, but some for pediatrics. This is an important column because it tells us that the vast majority of studies have focused on the treatment of vestibular schwannomas, which is incredibly important. But we also know that there are additional tumors which can develop in the setting of NF2-related schwannomatosis, such as meningiomas, ependymomas, and non-vestibular schwannomas. So we know that we're only really treating a small fraction of the tumors that can cause problems. And here are some of the results. I would argue at this point bevacizumab, or also known as Avastin, has been something that made its way into clinical practice in many places. The other drugs we use occasionally in the right setting, but have not had as broad an acceptance at this point. But this model here of one tumor type and one drug has inherent flaws that Dr. Bakker laid out. That is, it's too slow. It costs too much if you do it one after the other. And so we were looking for better ways to advance, treatment for NF2-related schwannomatosis. And this brings us to the INTUITT NF2 trial, which is a type of adaptive platform basket trial. Well, what is an adaptive trial? An adaptive trial is a trial which adapts or changes in real time. So I'll give you an example of how we force the trial to change in mid-lap, and you'll see why I think that makes our trials work better. A platform trial is a clinical trial in which multiple drugs are evaluated at the same time. And the way you coordinate multiple drugs is to have what we call a single master protocol that lays out the rules in which people receive these medicines. Finally, this trial is a type of a basket trial. And in that setting, these trials test the effectiveness of a single drug on different tumors that share a common genetic driver. So what does that mean? We know that schwannomas and meningiomas and ependymomas are all caused by genetic changes in the NF2 gene, and therefore we hope that one drug could have activity against multiple tumor types. So the rationale of the overall study is to create a trial that can rapidly and efficiently screen therapies for biological effects. And what's important is you can first look across all tumor types. You could say, for example, this drug causes 3% of tumors to shrink or 23% of tumors to shrink. You could also say, well, let's only look at vestibular schwannomas. Maybe there's a difference between how well the medicine works for vestibular schwannoma versus meningioma. You could also say, what about adults versus adolescents, or men versus women, or some other group like that? It gives you a chance to figure out who responds and what tumor types respond the best. The goal is therefore to move forward, if necessary, with any confirmatory studies that will allow us to get these drugs approved by the Food and Drug Administration. Here's the same graphic that Dr. Bakker showed. The top gray panel represents the master study, and the bottom gray panel represents the individual drug sub-studies, the individual drugs. If we first start by looking at the master study, this is what we call an allocation procedure. What it does is it allows a person with NF2-related schwannomatosis who has tumor growth to enter the trial. It then allows us to sort whether that person will be enrolled because the tumor growing is a vestibular schwannoma, a non-vestibular schwannoma, a meningioma, or an ependymoma. That individual then could get randomized to different medicines. Now, randomized just means it's by chance whether they get treated with drug A or drug B. And in my perfect fantasy world where I live, we would have 10 medicines at the same time we were studying. We don't have that just yet, although that's our vision. So we haven't had a randomization because first brigatinib was open, and as you'll hear about neratinib, so it's been more in line with each other. Anyway, after being allocated to one of these different sub-studies, a person then gets treated in this bottom gray panel, and they get treated as long as the tumor is stable in size or is shrinking. If the tumor starts growing, the target tumor, we interpret that to mean that the medicine's not working, that drug sub-study becomes discontinued, but that individual patient can now cycle back into the master study and await treatment from a new medicine. The goal is for a person who is looking for experimental treatments to be able to receive multiple over a short period of time. That is our goal. I haven't copied the map that Dr. Bakker showed, but here again is to acknowledge the amazing sites that are here that have worked on brigatinib: Johns Hopkins, Mass General Hospital, Mayo Clinic, New York University, University of Miami, and UCLA. And we hope to welcome more sites, such as University of Indiana, that will be starting with us soon. Okay. So here is how the master protocol works. We're accepting up to 80 eligible individuals. I already mentioned there's no treatment here. This is just a way to bring people into the program and allocate them or send them to different treatment sub-studies. We are going to collect natural history data, and we hope this will be open for more than 10 years. Well, what kind of characteristics are acceptable on this trial? Well, you have to have NF2-related schwannomatosis. You have to be at least 12. You have to have a type of tumor that's growing within the previous 36 months. That tumor has to be measurable by MRI scan. For reasons that have to do with the NIH and the FDA, you have to have a life expectancy greater than 1 year. You have to be able to care for yourself. Well, what would prevent somebody from joining if you're on chemotherapy or receive radiation within the last 3 years? If you can't tolerate an MRI, you have some illness which makes it unsafe for you to participate, or if you're pregnant or lactating, all those would not make you not eligible. Well, what are we looking at to determine whether these tumors are shrinking or, whether the drug is working? We look at what we call target tumors and non-target tumors, and this is something that's assigned by the treating physician. The target tumor, and there's only one, is the tumor that's growing that we use for the assessment of whether there's growth or shrinkage of the tumor. And that's because you have to have a rule, and you make a rule based on one tumor. But because persons with NF2-related schwannomatosis can have multiple tumors, and because we don't want to waste that information, we're also going to track up to 5 non-target tumors of interest. Our goal is to measure what is the tumor shrinkage rate, that is, what tumors shrink 20% or more. That's our primary endpoint. And our secondary endpoint is to look at tumor shrinkage among all the tumors, to measure the time to grow for each tumor. What is the rate of hearing improvement? What is the change in quality of life measured for NF2 persons? And what is the safety and the tolerability of these new medicines? I want to bring forward now how we're doing an adaptive model. I told you about platform trials. I've told you about basket trials. And now I'm going to show you what it means to be adaptive. We identified brigatinib as a medicine through the SYNODOS program that Dr. Bacher mentioned. It is an approved medicine. The fancy term is it's a selective ALK tyrosine kinase inhibitor. We know that it inhibits many of the growth signals that occur in tumor cells. It's approved by the FDA and in Europe for treatment of lung cancer. It's taken by mouth once daily. In the first stage of our study, we enrolled 20 participants, and we made sure that at least there was 2 participants for each of the 4 tumor baskets. After 6 months of treatment, we then did an interim analysis. If there were no responses, we were going to stop the study for brigatinib and say it wasn't active. But if we had at least 1 tumor that shrank, 1 target tumor, we were going to enroll 20 more people. But the key here is instead of enrolling 20 participants to all 4 baskets, we had an adaptation. That adaptation was we were only going to enroll those 20 persons into the 2 baskets that were doing the best. We wanted to increase the chances of finding a successful signal with this drug. So how did we do? This is part of the acceleration where we met our study accrual goals and timelines with enrollment from the first patient to understanding the primary endpoint within 36 months. And here is an example of how we did that. The red line here, what you see on the x-axis is time starting in June 2020 and going to August 2022. On the y-axis is how many persons were enrolled. Remember, our goal is 40, which is up here. The red line is what we thought we can do. We would be accruing people at this rate. When we started, our actual accruement lagged behind what we thought, and that's because first Mass General opened, then the second site opened, NYU, then the third site. So we were slow to open. We did our preliminary analysis. It did meet the criteria to continue. We had to let the data mature and analyze it. And then we beat all expectations in terms of accrual. So we accrued faster in the second half. And so we were able to finish that accrual in just 8 months. And we had this primary endpoint available for the CTF meeting earlier this year. So this represents a very fast accrual to the largest NF2-related schwannomatosis study to date. Oops. So here's some data here about who was enrolled. You can see we had 40 participants. The average age was 26 years, as young as 12 and as old as 54. We now have data on 165 tumors, again, showing the strength of the data set. That includes 40 target tumors And as I mentioned, we looked at additional non-target tumors, 125. And if you look here, our final data set includes 47 vestibular schwannomas, 56 non-vestibular schwannomas, 57 meningiomas, and 5 ependymomas. So I'll show you data in a second on some of this, but to say we have a much more robust data set at this point than we have for any other study. About a third of our participants were male, about a third were children or pediatric, 21 or under. The functional status, which is how well you can take care of yourself, was very high for our participants. This was a group of individuals who had had multiple treatments before, which makes sense if you're willing to go on a clinical study. 93% had had surgery, 23% had received radiation before, and almost 60% had received chemotherapy of some type. So this is not, for example, a person who had been recently diagnosed. These are individuals who had tried other things and felt like they needed some new treatment. So here's the big punchline for us, which shows you the change in tumor size for the target tumors. The way you read this, each bar here is a target tumor. Here on the y-axis, above the horizontal line is tumors that grew. Down below are tumors that shrank. And you can see the color coding here for the different tumor types. Out of 40 target tumors, we have 4 that met the bar for shrinkage. Now, when I say met the bar for shrinkage, you can see many other tumors shrank, but they didn't reach 20%. So if you look at the kind of overall result, about 10% of the tumors shrank. That includes 22% of the non-vestibular schwannomas, about 10% of the meningiomas. Unfortunately, none of the vestibular schwannomas and none of the ependymomas shrank. And that's why we knew in this data that the two groups that responded the best, the two tumor types that responded the best, were the non-vestibular schwannomas. That is, these are schwannomas that don't occur in the vestibular nerve. They can be in the brain, they can be in the spine, or they can be in the periphery. That these two groups were the ones that, for target tumors, responded the best. You can see here, this is the growth trajectory where they were growing. This vertical line here is the initiation of treatment. This is the growth pattern before treatment, and this is after. And what you can see here is we have some flattening out of the curve. Not every tumor flattens out, but we're seeing a significant slowing of the tumors before and after treatment. So there's two effects. There's this idea that we have tumor shrinkage, which is important, but we also see here that there's a time to delay treatments, which in some persons meant they didn't have to get surgeries for many years. And so that's a positive also, and we're exploring that in subsequent analysis. How about hearing? This is a way to look at hearing. We have better graphs now, but you can see if you're looking at word recognition score, this is the percentage of words you get right, whether you got 20% right or 60% right or 98% right. If you look here, purple is hearing loss, orange is stable hearing, and green is hearing improvement. About 35% of ears, if you look at ears separately, left ear, right ear would be 2 ears, 35% of ears had some improvement. If we look at hearing as a measure of pure tone average, how loud does a word have to be for you to hear it, or how loud the sound has to be? Again, with same code, about a quarter of ears got better. That is more sensitive to hearing. So we did see hearing improvement in this study. Overall, the medicine was well tolerated. This is a lot of medical legalese, so I didn't feel like it was worth it to go translating. There was no serious or there's no life-threatening or adverse events or side effects, and there was no deaths, no serious adverse events. And what you can see here is the GI symptoms were the ones we heard most about. We do see some laboratory abnormalities, which in general were well controlled. And we did see some individuals who had cramps, high blood pressure, fatigue, and so forth. So we don't want to poo-poo the side effects. They're real, but for most people, they were definitely manageable in the long term. So where are we now? What's the next step? First of all, we believe that these initial results demonstrate that there is clinical activity of this medicine, particularly for non-vestibular schwannomas and meningiomas. We acknowledge there's less benefit for vestibular schwannomas and ependymomas, but I want to be clear, I don't think there's no benefit. I think there is some benefit for some persons. We did see some non-target tumor shrinking. We are continuing the analysis of tumors and hearing outcomes. And one of the things that I'm very interested in pursuing and would like to report soon is the difference in children and adults. We know that sometimes children have a lower response rate compared to adults. We're taking a deep look at quality of life on the medicines because we know it has to be tolerable. And probably most important is Dr. Bacher and I and others have ongoing discussions with Takeda to say, can we take this medicine and combine it with other medicines to form a cocktail? Because that approach is what we do in cancer treatments. We recognize that it has to be tolerable, but we're very interested in moving that forward. This is the last slide before the acknowledgement. So how are we accelerating research? Well, this design has a lot of advantages. We rapidly accrued 40 participants, the largest trial. We determined the activity of this drug in 4 different tumor types. Just to be clear, that's like running 4 separate trials in one. We also identified which tumors are most likely to benefit. And then we also did this in a way that was cost effective. The good news is we're already deep into our second study with neratinib. And thank you for those individuals who are still on this trial as well as brigatinib. We have 11 of our 20 participants in our first stage, including 2 participants who've previously been treated in brigatinib. So that's our goal. And then we are going to be meeting with the FDA about this design to see how do we get this to what input do they have for us to make this a vehicle for getting drug approval. And with that, just to acknowledge the team, the large team and individuals from across the country and the world, really, that helped make all this happen. And of course, a special thank you to all the patients and families who are involved in this research. And with that, I think I'll turn it back over to the rest of the speakers.
Hi, everyone. I'm going to now take you through the final stage of the talk. Just want to bring my slides up and I'm trouble finding oh, here it is. There. So good evening, everyone. As was mentioned, my name is Bridget Gerlich, and I'm going to take you through the final stage of the talk tonight. As we heard from Dr. Bacher and Dr. Plotkin, how can we really build on our success and continue to accelerate the path to approval? So really, what's the next? As we heard from Dr. Bacher and Dr. Plotkin, what we did has worked, and we recognize the limitations of rare disease in the traditional trial sense, and we want to now expand upon this. So the first part, as Dr. Bacher spoke to, is preclinical testing. We know that using the right platform and assays for preclinical data can effectively predict success when we enter the clinic. For NF2 ready models and cell assays, as Dr. Bacher pointed out, this is a rare disease and not everyone has access to these. So CTF is building on that SYNODOS model, is looking to will be creating a preclinical hub. And within this hub, the central site, it will include a team of experts that will work with our stakeholder partners to assess each asset and drug as it comes in and determine which model systems and which cellular assays and other aspects would be necessary to best predict if this drug was relevant in the NF space and moving into the clinic. So what the hub will include will be a menu of pre-selected assays that are all in place. And there will also be agreements with all our hub partners in place. So as was also pointed out earlier, the delay to setting up these models and negotiating the contracts with the academic centers will no longer be an obstacle. And our aim is then to attract more pharma partners to investigate their resources and their assets in NF research. And as I pointed out, and most importantly, there will be an expert panel of those experienced, both clinical and preclinical NF stakeholders that can effectively guide in the right path for preclinical assessment. So where are we with this preclinical hub progress? The first partners have been identified. We have one MSA in place from an academic lab. We've also, in the last several weeks, set out an appeal to academic labs to join us in this public-private partnership that we're embarking on. And we're outreaching to all the known private partners that we're aware of with exciting models and cellular assays and asking them to join our movements. So what do we still need? Funding. It's so important, and it will take money to set this up and implement it. And once activated, the goal is to make this a self-sufficient model that as partners, particularly from the pharma industry, come in and assess in this space, they could also contribute. So it would be shared expenses, and it would be a sustainable model. I'm not going to spend too much time on this. I think we heard quite a bit about the limitations of standard trial design, particularly in rare diseases, and what the bottlenecks are, and how a platform basket adaptive trial can really expedite moving identified assets and molecules through a clinical trial. But also, how can we expand on what we're doing in INTUITT and provide access to even more patients, and also make more drugs available to both NF1 and NF2 patients? In order to do that, we've recently partnered with GCAR, the Global Coalition of Adaptive Research. They're leading experts in managing drug-related platform and process trials. They bring with them the leadership and also the regulatory expertise and expertise in site management. They are known for their success in the Agile trial and brain tumors, and we're really excited to be now working with them. And we will work with them as the global leaders in neurofibromatosis and help bring together patients across the globe. And I should mention that this will be a global initiative in both transatlantically in both Europe and the US. Eventually, as we plan to begin one trial in Europe, in NF1, the hope, and we're already working with our United States partners and the trial consortium to ensure we're aligned on this plan, as it is important to bring all stakeholders to the table early on, and patients as well, who were very involved in the trial design. And for NF2, we plan to partner with our good friend Dr. Plotkin and his INTUITT platform, and go from there. So where are we with the platform basket trial, that the NF1 trial that we're beginning with to test this model? We have the master protocol in place. I should also state that we do have a master protocol in place for NF2, which we will be looking to implement once we establish and know the feasibility and success of the NF1 trial. Partners have been identified. We've had conversations with all of our primary investigators across Europe, and we're establishing a committee structure. So what's next? Again, the importance of securing funding in order to the startup costs, in order to organize the trial, get regulatory feedback, and begin implementation of the master protocol, as Dr. Plotkin eloquently pointed out, where patients will be on ready to be treated. We expect that will fund, together with GCAR, we'll have to fund that part of the project. But the long-term aim is to have this also a self-sustainable trial that, as the success is demonstrated in this rare disease population, and that how it can accelerate, and with adaptive design, you can quickly identify a go, no go situation. It will attract more pharma partners, and also and they will bring with them their assets and implement and share in the sustainability and funding of the project. And with that, I want to thank you and not take up any more time, because I know there's a lot of questions from this exciting talk that Dr. Plotkin gave.
Thank you so much, Dr. Gerlich, and to all of our presenters tonight. I really appreciate everything that you've brought to us. I also want to just say quickly that Dr. Plotkin has really graciously been answering a lot of questions in the Q&A. So I am going to ask some of those now of our panelists, but there are also some great answers in the Q&A. If you look at the answered questions, you should be able to see that. So to start, I'd love to bring up Barbara Franklin's question, which I know you responded to, Scott, but for everyone's benefit, Barbara asks, what if the drug impacts a different tumor than the one that is selected initially to watch? What is the result? What happens then? Sorry, Dr. Plotkin, I'm talking to you. He's being so good. He's in the Q&A answering questions. That's why.
Can I just finish? I was just give me one more second.
That's totally fine. No, that's fine.
Sorry.
No, it's fine. All of our questions are mostly for Dr. Plotkin, which is fine and makes a lot of sense because he brought some really great information to everyone tonight. So thank you for being here. Okay. Also, I'll say quickly while he finishes up that please, if you have anything that doesn't get answered tonight and you have any concerns, I'll put my direct contact information into the chat right as we're about to close up for the evening, and you can always reach out to me directly at any time. Okay. So what I had said, Dr. Plotkin, is you had actually responded to this in the Q&A, but for everyone's benefit, if they aren't looking in there, Barbara Franklin asked, what if the drug impacts a different tumor than the one that is selected initially to watch? Yeah,
that's a great question. Barbara always has good questions. She has lots of questions for me. That's why we're looking at them all, right? The FDA requires us to have rules. We're not allowed to be free agents. We have rules. This is put down in what we call a protocol, which is a formal document that's approved by our ethics committee, approved by the FDA. Those rules outline how a person on the study is treated if this tumor grows or that tumor grows. So by selecting a target tumor with the treating physician, that individual will help select what's the target tumor. So let's say if you have a vestibular schwannoma and a meningioma, they would usually choose one that's growing the fastest, so one that is most important to address at that point in time. That tumor becomes the target tumor. But to take Barbara's question, let's say the vestibular schwannoma continues to grow slowly, but the meningioma shrank. Of course, we're happy about that, but we have rules. And if that vestibular schwannoma grows more than 20% compared to baseline, we are required to have that person get off of this brigatinib study. So, of course, we would all be disappointed when that happens. But to be clear what we're doing, brigatinib is an off-label medicine that sometimes you can get approved if there's evidence that it was working for you. So in that situation, just so people understand the clinical context, many of us have been successful appealing to insurance companies to say, listen, this drug has worked in a clinical trial. They can't receive it anymore according to the rules, but we believe it is providing clinical benefit. So that's how we would try to, I think, deal with an issue like that.
Oh, I'm muted. Thank you so much. So then Michael Skyer asks a question of how are new drugs or new baskets identified?
New drugs, not baskets. So the baskets are fixed right now. So there'll be the 4 basket types. So thank you, Michael, for that question. He's also full of good questions. This comes from our amazing preclinical and clinical research group. We talked about SYNODOS, which was this very large multi-institutional study to identify active medicines. But outside of SYNODOS, we have amazing researchers. So our team, first of all, we're completely open. Any group in the world that has a preclinical data in a test tube or in an animal model of NF2, if they have data, they can come to us. We don't care where the data comes from, but we need data to support it. So if there is data suggesting that a drug can work, I will talk to any company on the planet Earth to track it down. Some companies are able to work with us and some are not. And as you can imagine, there has to be safety data. I think at this point, this trial is not designed to take a medicine that's never been in a human being before, because that's called a phase 1 dose finding study. We're only taking medicines that have safe treatment levels. So that would be we would still study a drug like that, but in a different setting. So that's really how and we've talked to probably 20 companies already. Some of them are able to participate and some of them are not.
Excellent. Thank you so much. We also had a question from an anonymous person, which is fine, that I thought was interesting, asking about saying not to oversimplify, but are these results saying that Avastin is still a more effective treatment for the vestibular schwannomas than brigatinib?
Yeah. So I try to nuance this a little bit. I hope I did a good job, which is it is complex. So I agree with your statement that we don't want to simplify it too much. The people that were enrolled into our previous studies with bevacizumab, Avastin, are individuals who had to have growing vestibular schwannomas and hearing loss. That is a different group of individuals, a different set of requirements than we had for this one. Having said that, my own personal feeling is that bevacizumab remains the gold standard in terms of treating hearing loss. And yet I also would say that the medicine brigatinib has been more tolerable for more people and easier to access because it's a pill you take at home and you don't have to travel to a specialty center to get treated every 2 weeks or every 3 weeks. So I would really frame it more as you want to choose the right medicine for the right scenario, and you'd want to talk to the physician to see which one. I can definitely see an instance where it might be worth trying brigatinib once we have the data out. I'm not suggesting we rush to it now. We don't even have our full analysis set yet, but I can see a scenario where an oral medicine could be better than an IV medicine. So complex.
Excellent. Yeah, thank you. I think we have time for maybe just one more. So our most popular question with our upvoting process is, do we know if patients will need to take treatment for life? And do we know what happens when treatment is stopped?
So those are complex questions, but very good. I'll answer in the general first. I'll say cautiously that I do think people will need extended treatment. Lifetime is a big statement, but I think I would caution people in my clinic to say that if we have a medicine that's stopping your tumors from growing, I think the most reasonable to think is you're going to be on it a long time. So your doctors, our team, all the doctors here are trying to think, how do we make medicines tolerable? So I'll give you an example. Some people that I treat, we do Avastin 3 months on and then 3 months off so that there's a treatment break. We're very interested in understanding for brigatinib what happens when people stop. We don't have that data yet. And I think the question might be, is it possible to start and stop? That kind of research, I think, comes after we determine whether a medicine works or not. So our first step is going to be, does brigatinib work and in what setting? And then once we do that, we're going to start getting some real world experience and designing follow-on studies. For example, maybe you can do brigatinib 1 month on, 1 month off, or 5 days on and 2 days off. How do we make this work for everybody? We know we need to do that. And I think that's going to be step 2 if we can get the FDA to put a stamp of approval on this.
Excellent. Thank you so much, Dr. Plotkin, and to all of our panelists. As we're
Maybe I
nearing
wanted
the-
to add one
Yes,
quick thing
please,
here,
of course.
Kate, is that people may have wondered, I can imagine, that why the heck would we make pig models for NF, right? We have made those pig models for NF1. We have tried to make those pig models also for NF2-related schwannomatosis. And the reason why we are interested in that and looking at maybe opportunities for the future is because if you want to study exactly what Dr. Plotkin said, right? If you want to know, can we lower the dose? Can we increase the dose? Can we play with this? Can we play with that? The pig models are in fact excellent because they have very similar metabolism as we have. And it really will help us to do all those what they call pharmacokinetic and pharmacodynamic studies, right? Looking at what does the body to the drug and what does the drug to the body. So those studies could perfectly be run in a pig model much better than in a mouse model because mice are very different. So just wanted to add that by why pig models.
Excellent. Thank you so much. So again, thank you to all of our panelists. And as we are nearing the end of our time, I'm really pleased to finish out our evening by introducing our CTF board member and NF2 parent, RB Harrison.
Thank you all for joining us, and a big thank you to Annette, Scott, and Bridget. It is wonderful to see how the collective investment of our donors, friends, and families have helped CTF bring drugs to clinical trials. As a board member of the foundation, and most importantly, an NF2 parent, I'm personally vested in CTF's vision to identify, test, and place in clinical trials drugs like brigatinib that we heard about tonight. And in fact, my son is in the brigatinib trial. Every day, I watch the challenges all of us in this call face from NF. I personally invest in CTF's mission because I believe CTF is the catalyst for a change in research that will get us new treatments and ultimately cures faster. Tonight's program made my belief even stronger. The reality to achieve this vision, we need funding. I hope you will donate tonight or before year end as we work to raise $130 million over the next 5 years to ensure that our patients have the treatments they need. You can go to ctf.org/donate to make a donation or reach out to donor relations at ctf.org. Thank you again very much for attending, and I hope you will join us to defeat NF.
Thank you so much, RB. That link is in the chat for everyone. And thank you again for joining us.